All right, thank you everybody. Thanks for the organizers for inviting me. It's a pleasure to be here at this conference. Really enjoyed what we've heard so far. Looking forward to give my talk and present you some ideas around my own personal journey with IBD and SCD. I'll talk a little bit about the microbiome underlying what we're all doing. I'll give you some examples of the food interacting with microbiome, interacting with SCD, how that plays out with travel. I'll give you my own example of what happened to me when I traveled and the microbiome influence. I'll talk briefly about the gut-brain relationship, so microbiome interfacing beyond the gut. And then I'll have some closing remarks. So before I start, just a quick background on myself. I'm out in Seattle. I've been there about 20 years, lived in the Bay Area, San Francisco for about 20 years. I studied out in UC Davis, got an undergrad in genetics and a PhD in nutrition. Finished in 1998, and then 3 years later I was diagnosed with Crohn's. Probably had it about 15 to 20 years at that point, but it wasn't diagnosed. Kind of struggled with it throughout that period, on and off, without really knowing. But probably in the back of my mind, it just made me attracted to nutrition, and that's probably why I went into that field and got a PhD. Didn't really know why. So here I am in 2001 with a diagnosis in my ileum. And right away, because it was so late in the disease course, I had to have a resection. So about a 30 centimeter terminal ileum resection. I was already on mesalamine. Obviously, that was too late. Prednisone was too late. Then went on Remicade, Aza, Tysabri, Humira, the whole sequence. So every few years, I would have to change. Something you probably know about. 2005, I had to have another resection. So at this point, a lot of my ileum has been removed, and my doctor suggested that I should try Entyvio. Entyvio failed after a year, or I didn't respond to it. And finally, I was presented with Stelara, which has just been recently approved. And at this point, for the first time really in this entire sequence, a light bulb went off in my head. I had, up until now, compartmentalized my disease, left it up to my doctors to handle for me, and then I lived my life over here. IBD was over here. They never really crossed over until I was presented with Stelara because it was the last in the line in the pipeline. And asked my doctor what happens if Stelara fails, and at that point there was no other option other than going experimental. So really, I felt I'm sort of over the cliff, watching myself about to fall, and I had a big epiphany. I realized that here I am as a scientist. I've never really dug into this myself. What is going on? I dug up the paper on Stelara, and I just knew it's not going to work for me. Just the efficacy and the response wasn't there. So I asked my doctor to pause. He put me on prednisone until I could figure out what I want to do. I quit my job, came home, and literally dug into everything I could find. I'd already known about SCD, but just never really committed myself to it. So here I am. I decided to do SCD as my standard remedy. I was placed on 40 milligrams of prednisone. I'd always responded to prednisone, so that was basically my insurance policy. And I worked out a partnership with my GI doctor. Initially, he didn't want me to do it, didn't know about it. This is in February of 2017. But I worked out closely, partnered with him so I could do this, and he would watch me, but a little bit at a distance. So I kick off SCD, and today I'll talk about quickly becoming symptom-free, and for the first time in 18 years, medical-free, medicine, no pharmaceuticals. Okay? So what happened here? So 2016, post-surgery, this is my second surgery, I signed up to this program in Seattle out of Institute for Systems Biology, a company called Arjomand. This program, it's a scientific wellness program. They give you coaching, some nutrition advice, got a bunch of labs measured over and over again, microbiome. I had my entire genome sequenced, and I had sort of light touch coaching, not IBD specific, certainly not SCD, but it was a sort of generic measurement over this period. Within a year, I knew this isn't working at the degree I needed, so I jumped into SCD and kind of managed it myself, launched it, and implemented it myself. And I won't go into the details, you know all about SCD. But I did it quite strict, by the books, and stuck with it in the kitchen 4 or 5 hours a day for 6 months, ate nothing but breakfast, snacks, lunch, everything. Rejigged my entire kitchen, got a separate freezer, refrigerator allocated to SCD. I'm the only one at home that does it. And I became pretty good at it. It's kind of fun now, right? So let's look at what happened with my labs, just because I happen to have a whole slew of labs before and after. This is my cholesterol before and after SCD. So that last measurement was made 6 months into SCD. Everything else was made prior to SCD. Pretty well managed. There isn't a day I don't eat 3 eggs. And I mean, cholesterol is right there on the bottom. I'm borderline low on cholesterol. All right? Yeah. LDLs right at the bottom. No issue. SCD, in fact, probably makes it even better. Triglycerides are a bit on the high side. This is, mind you, after being on steroids for about 8 years. They tend to affect triglycerides, and so that's probably why it's at the top. Blood pressure, talk about, you know, cardiovascular, stress, you name it, was pretty low, went even lower on SCD. Okay. Toxins, we talked about this. I had my mercury measured before and after. Yeah, look at mercury. It goes up a little bit on SCD. Still in the acceptable range, but I attribute this to my increased consumption of fish. I was really focusing on having good fish, and I have sardines and canned wild fish. So this is something that we may want to watch on SCD. Just have your mercury measured if you're concerned about your consumption of fish or fish oils. Lead wasn't an issue. My folate, some nutritional markers, all good. Zinc went up. I was supplementing on zinc, so you can see it's responding and going a little bit over. So you can't actually overdo supplementation, so keep an eye out. B12, I needed heavy supplementation on B12, oral, not sublingual. Remember, I've had terminal ileum resection twice. That's where you absorb B12. So I had to really keep an eye on it, and I kept my supplementation the same throughout. But interestingly, on SCD, I was able to bump up my B12 levels. And to me, this means I was actually able to absorb more of the B12 in my terminal ileum because of the healing. Okay. D, vitamin D, we've talked quite a bit about it. While I was on the Aravail program, I was struggling to get myself up above 30, and the goal line is 40 actually for IBD. So we sequentially kept increasing my supplementation to 5,000, 10,000, finally 15,000 IUs a day is what it took for me to get to above 40 nanograms per mil. Now, I do have a SNP. Part of my genome sequencing showed that I have a bad vitamin D receptor, and it impacts my ability to absorb and take up vitamin D. So here I am. I have to kind of overdo it on this side. And I have to keep going. And if I don't keep an eye, I might drop below 40. So I have to kind of keep an eye on this, make sure I'm above 40 nanograms per mil. It's been shown to be protective. Albumin, again, more sort of status markers. Everything's in check. It was in good order before, after. Hematocrit, always been on the low side, but here I am, pretty much in the good range. Diabetes, okay, this is a big one for a CD, right? All that glucose, right? All the sugar. Glucose was always, fasting glucose was always in the good range for me. Insulin dropped, which is a good sign. Doesn't have to work hard because there isn't a whole bunch of sugar coming in. And hemoglobin A1C, dramatic drop. All right, this is a long-term marker of glucose exposure. Okay, so these are really good indicators of how sugar removal actually has non-IBD related benefits. Obviously, it's going to improve the inflammatory aspects of sugar, but you're going to get secondary benefits on diabetes and other factors that are influenced by high sugar and sort of pre-diabetes. So another key point. I was, again, heavy on sort of trying to keep the good lipids. This is an omega-3 index, checks out quite well. Now here are some inflammatory measurements, right? Here we go, IBD. Right before I did my SCD, I was really on the high end. This is what scared me. This is when I had that epiphany and my doctor said you should switch to Stellaro. Interleukin-6 is an inflammatory marker. It was right there on the high end, dropped dramatically on SCD. CRP on the high end, dropped dramatically on SCD, right? These are all good indicators of response. Interleukin-8, another marker of inflammation. It was low. Unfortunately, the labs weren't geared to measure it at that midpoint, but it got a little lower on SCD from this earlier time point. 8.0 went down to 3.5. And TNF-alpha, right? This is a target for a lot of the biologics that I was on. Even on Humira, I had a 4.1 high level of TNF-alpha. Wasn't doing anything. I was inflamed, putting out a lot of TNF-alpha. On no medication and an SCD, look at where the TNF-alpha is. Okay? That's an analytical confirmation that I wasn't putting out inflammatory signals. Right? Good signs. Now, fecal calprotectin, we've talked quite a bit. I was at 493 before SCD, and look where it's at now. Okay? Consistent drop, steady. I have to get another one in a few months, and I'm sure it's going to be good. So, what about the microbiome? I was giving stool samples, and they were tracking it for me. So here is the entire population of that Arjomand program, that wellness program I told you about. Roughly 10,000 patients, self-selected, so a lot of them are healthy, some might have other conditions. This is the general spread of diversity, and this is where I was. Okay, way on the low end of diversity both times. Without SCD, it just wasn't bumping up. And even with SCD, 6 months into it, actually 11 months into it, hardly a noticeable movement in the score. Okay? Now, just keep that in mind. These were measurements of phyla, so at a fairly high classification of microbes. So at the phyla level, I wasn't getting a whole lot of fluctuation. Okay, so I'll talk about that. So when I was at Bacteroides, this is not necessarily the friendliest phyla, kind of stayed the same, with and without SCD, with and without other interventions. Fusobacteria, not a friendly phyla you want to have. I'm really high. I was on the highest one in that entire Arjomand population. Okay, doesn't move. Maybe this is too soon. I'll talk about some of the possibilities of what might explain that. So now, what happened about some clinical markers? This is my body weight throughout years and years, actually. SCD started at that point. I've lost about 15 pounds since starting SCD. I was 165 roughly when I started. 160, and I'm 145. But it's about 15 pounds higher than my lowest point. So sort of in a pretty good zone. I'm not worried about it. I keep an eye on it. I think it's a natural side effect of being on a good healthy diet, being off steroids, right? And I just keep an eye on it. I tend to eat about 3,500 calories a day to maintain my weight, 4,000. I might gain some. But again, I don't have a whole lot of ileum, right? So that's kind of where it is. So to sum it up, after 2 weeks, I lost all my GI symptoms on SCD. After 2 months, all my joint pains went away. Began doing yoga routinely 3 times a week. And I'm on 0 meds at 6 months. All right? After 3 years, still no flares, no hospitalizations. That was a regular occurrence. No medications. And then the drop of body weight I told you about. So this sort of sums it up of where things are. My GI doctor was amazed, right? After 6 months, he said, Ali, what's the name of this diet you're on? What's a typical breakfast? What do you eat for lunch? You got to come back and tell the other GIs about this. Can I set up a presentation for you? So quite a turnaround. He has terminated my tapered off prednisone at 6 months. At 1 year, he had me come back and do a colonoscopy and MRI because now wash out all the effects of prednisone perhaps. So here's my pre-SCD on prednisone, on Entyvio, MRI, and colonoscopy results. Fibrostenosing disease, 14 centimeters in the ileum, active inflammation adjacent, distal ileum, and for the first time, I was getting inflammation in the jejunum. I'd never had that. Okay, this is what launched my SCD, scared me. A year later, 6 months after stopping meds and prednisone, that 14 centimeters had shrunk to 5 in the ileum, no generalized bowel obstruction, and resolution of the prior jejunal inflammation. Like that's as clear as it gets in terms of what you want to see, actual resolution. So yeah, this was good. In the meantime, I was enjoying all the foods you like, right? All the things you know. It became pretty good. That's breakfast. Lots of eggs, omelets, concoctions. I just whip things together a lot of times. I don't need to plan too much ahead. Make broth when I want to. Lots of grilled fish. Make my own pesto. A lot of nice salmon we have out in Seattle, right? Can't get better than that. I just sort of modify some recipes. I follow the recipe books. They're always great for ideas. Yeah, a lot of good stuff. Snacks, cookies, make my own almond milk. And then work, right? I started going back to work and kind of keeping it balanced 3 days a week just to kind of not offset the gains I'd made. But I would pack lunch, right? Pack lunch, pack this, pack that. It's pretty easy. Yogurt was a big part of all this every single day. Here we are. So I'm feeling better. I'm back to work doing yoga. Actually started growing my own vegetables. Here I am, raising and growing and eating lots of greens. I had probably more fiber in my first year on SCD than I had in the prior 16 years combined. All right, and that's literally how it shapes up. My wife would see me eating this salad and just turn around and walk out the kitchen. She's like, I can't watch you do that. And then she realized that's actually the solution, right? It's just counterintuitive sometimes. So here's my vegetables and so on. So now let's take a closer look. So here I am getting well, lots of time in the kitchen, stuff baking in the oven. Of course, I go online and try and figure out what just happened, right? Scientifically, I'm curious, right? I should be able to explain some of this. So it turns out there's a lot of science known and a lot of pieces came together, and I want to explain some of the things. So as you might expect, the microbiome is a big hot topic. Just between 2013 and 2017, there's over 12,000 publications. That's roughly 80% of everything that's been published in the last 40 years happened in that 3-year window, 4-year window. So there's just this big kick up in microbiome research, microbiome publications. If you look at the word cloud of the top cited papers, this is sort of the most common set of words you see in these papers. Gut microbiome, right there at the top, inflammatory bowel disease. All right? So this is a pretty key area. Now, to give you a quick overview of what we're going to talk about next this is a schematic of the GI tract, and the gut microbes are laid out vertically, top to bottom, and increasing in density as you go down, quite dramatically, right? So the stomach's pretty much sterile, 10 organisms per gram, right? Shouldn't be up there. And as you go down, it increases to 1,000 per gram, 10,000, terminal ileum, 10 million, okay? And then suddenly a big kick as you go to the colon. So that's sort of the vertical geometry. The horizontal geometry, if you take a slice of that anywhere in the GI tract, they're mostly concentrated in the lumen. Okay, so the vast majority sit in the lumen, as you might expect, and then some smaller amount hang out in the mucus layer, and very, very little, if any, hang out at the epithelial surface. So that's sort of the horizontal geometry. And of course, there's a pH gradient going down into the stomach and back out, and an oxygen gradient that's sort of important to keep in the back of your mind. So some of these studies look at all this, and what do we get out of it? Now here is a study in non-human primates looking at stool measurements. Here are 15 monkeys looking at their stool output and the kind of phyla they have between them. That's over here, spread out one by one. Now, if you look at the lumen, we had access to the lumen. These are animal studies. This is what the ascending, the ileum, the ascending, transverse, and descending composition looks like. And then if you go down to the mucus layer, remember that outer ring, this is what the composition looks like. So you can see that what you get in the stool isn't what you have in the mucus. Not in the jejunum, not in the ileum, nowhere. There's a mismatch. Okay? So if you're collecting and doing microbiome analysis in stool, is it reflective of what's going on in the mucus? So here's another look at it. This is what gets picked up in the large intestine and in the stool. Is there a comparison? Is there a concordance? If you lay this out, it's pretty good. The blue dots are, there's gray dots. You may not be able to see it. It's pretty much a straight line. So in the lumen and the stool, there's pretty much a straight line. So it's good concordance. But if you look at the lumen, the mucus, you start getting some spread. All right? So again, the stool doesn't necessarily represent what's in the mucus, and it gets even worse in the small intestine, a lot worse. Right? So the concordance between what you pick up in the lumen and in the mucus of the small intestine doesn't really predict what comes out in the stool. So if you're sampling in the stool, you can't make too many interpretations of what's going on in the small intestine, neither in the lumen or the mucus. So, does that explain what I observed, right? Remember, these are my phyla, didn't really change on SCD. So, do I just need more time? Let it take hold over a longer period than 11 months? Does stool sampling changes show up changes reflective of the small intestine, like I just showed you? So maybe all these alterations that I was creating with SCD was happening in the small intestine or was happening in the mucus, up and down, and it's just not getting picked up in the stool. Maybe I need actually a deeper presentation of the data. So all this data is phylum level, right? So at a phylum level, maybe you don't see changes, but if you go to a deeper granular level, that maybe just subtle changes there are enough to give you the results you're looking for. Or maybe it's just more complicated than this and we just don't know yet. Okay, so that's pretty interesting. Now, a few years ago, the NIH launched the Human Microbiome Project. Part of that was the Integrative Human Microbiome Project, looking at 3 specific disease conditions or life conditions: pregnancy and preterm birth, IBD, and onset of diabetes. I'll focus on the IBD side. These are, first of all, about 500 healthy subjects that were looked at the phyla level and just ranked from highest to lowest in terms of, here we are, bacteriorities, right? Just kind of length. And you can see there's a spread in the population, right? Kind of natural. You just kind of rank them one by one. Because this was a multi-omic study, we can also rank them by what these microorganisms are doing. What chemical pathways are they capable of doing? All those genes that they have and we don't. Now, if you look at these same subjects, these healthy subjects, what explains that spread? There's diet. Several factors were picked out. Whether they were breastfed, have ever given birth, all these other factors. And on these species, you can see which ones explain the uptick or the decrease, the red and the blue. So, for example, ethnicity happens to be the biggest factor right there, right? Why some of these 500 people had high or low levels. A lot of that can actually be explained by their ethnicity. The other ones that are pretty high up there are whether they were breastfed, whether they had given birth. But at any one of you, you can't really change that, right? It is what it is. You were either breastfed or you weren't. You're either, whatever your ethnicity is. But the one factor that you can change is diet. Right? And obviously, that's what we're going to talk about. So they did the study on the healthy subjects, then they moved on to IBD subjects. Right? And here they started looking at what's changing in the gut, what transcripts, what organisms are seeing at a higher level in IBD patients, and more likely, which ones are depleted, which is common, right? Like me, I'm on the low end of the diversity. We're probably all quite depleted on a lot of species. And of course, this translates to downstream effects. These upregulated or downregulated numbers of strains has an impact through the lumen, creating all these downstream effects, creating inflammation. All right, so that's the sequence. Now, if you want to develop an intervention that reverses all this, up here, it's a really complicated mess. There's a lot going on. There's no one single button you can push to reverse it, but they sort of all congregate and intersect down here at the inflammation level, right? All these, whatever's happening up there, it's sort of ending up in inflammation. So if I'm a pharmaceutical company, do I work up here in this sort of unknown, uncharted territory, or yeah, I understand much more on the inflammation side. So now that's why you have all the drug companies. Basically, most of the IBD targets are focused on this inflammatory reversal, sort of inhibiting the inflammation, not really looking at the upstream cause or the trigger. But that has its own limitations. I'm just going to pick on Entyvio here. These are the results of the trials that Entyvio has on, this is UC, trial 1. If you're looking at just improvement, let's say clinical remission, 17 versus 5 in the placebo, 40 versus 25, endoscopic appearance. I'll just flip these fairly quickly because you probably know all this. It just doesn't get you even halfway to remission, in terms of the population. They're always sub 50%, 40, 50, 30% response. This is in Crohn's disease. So there's this glaring apparent admission, really, that with biologics, you're not going to get to the finish line. You're not going to cross that line. It's maybe gets you halfway there in the population. And if you are responding, when do you lose response? Is it a matter of time, or is it a sort of a lasting response? And it's becoming apparent it's not. And so we need help. We need to fill that other gap, the other side, to get to 100%. And we talked about a bunch of things. We'll talk about diet. So here's the pipeline of IBD drugs. There's a lot of companies working on IBD drugs because they recognize that the current approved drugs right here are not cutting it. So it's actually a lucrative investment to come up with the next one because there's an opportunity for them to sort of cover what's missing right now. Now I won't go through all this at all, but notice that there's a whole channel of activity around the microbiome. So that's the upper part, that lumen that I showed you, not the inflammation. So they're moving upstream, looking at targeting, pushing buttons at the sort of source, at the trigger source. But look at what they're working on. Eradicating E. coli, adherent E. coli, citrus extract, flaxseed. This is sort of phase 2. Phase 3, there's some antibiotics here, fecal microbial transplant, curcumin, right? You kind of get the idea. They're moving away from these sort of xenobiotics, these foreign agents, and looking at sort of dietary interventions of some antibiotics. Now, notice there's nothing in the market here on the microbiome. It's a blank. But there is, right? I mean, that's where we are. We're already there. We were there years ago, right? So how's it working? What makes SCD work? And we're kind of already there in the finish line. So looking at IBD microbiome diversity, this is now IBD patients in the Human Microbiome Project, ranked by their diversity score. So I had my data earlier. This is the IBD, human microbiome population set. And in this case, they were just ranked from lowest. The green line represents the healthy plane. So your healthy population is over here on the green line. And the IBD population is a distance away. So how far away? Well, you can just rank them. Some patients are quite far away. That's probably me. And some are pretty close. So what else do we know about these? Why are these patients up here and some are down here? Well, we actually know a little bit more about their background. So let's color this chart. And look, it turns out that if you have an ileal Crohn's disease in the ileum and an ileocecal resection, hey, that's me. All those people are bunched up at the top. Look at that. Quite far from the health plane. The next group down is without resection, but still in the ileum. Next group down, colonic, all right, Crohn's disease. Then maybe some UC patients are here. And finally, the greens are the healthy controls. All right, so this is sort of this transgression from a healthy plane. If you have it in the ileum, it's probably bad. If you have a resection, it really pushes you out from the healthy plane. Now here's a three-dimensional representation of that same data. Okay? So all the dots, the yellows are ileal Crohn's with a resection, bunched up over here on a three-dimensional chart. Everybody else, the greens and the ones I showed you earlier. So that's very visual representation of how far we are from the healthy plane. But this is one single time point, one sample. Now if you do more than one sample, it shows you the fluctuations day to day or week to week. And look what happens. So all the other patient pools, the healthy, UC, non-resected, sort of fluctuate, but minimally, right? There's a little bounce. But look at the ones that had ileocecal resection. They're all over the place. It's a very bouncy, flexible, sensitive parameter. So any given day, you could have a very variable microbiome. And that's probably one of the factors we want to reverse to stabilize it, because that's what the healthy people show. All right? So this is just a schematic of all the influences that we can do. Typically, high sugar, excessive protein, saturated fat, taking protein pump inhibitors and antibiotics, and you get all these negative outcomes. And I'll go through some of these. And there's an opposite to this. There's a couple of things we can do that have positive influence. One is taking probiotic and prebiotic, right? So SCD, that's yogurt and fruits and vegetables, nuts and seeds, fiber. And then you get all these positive outcomes. So it's a pretty thin line between this and that. And again, for 16 years, I was told eat a low residue diet. Probably before that, I wasn't really eating the best diet either. And almost no fiber and probably no fermented foods. So I was depleted, right? Now, what do we mean by the specifics of these foods? This is a quick schematic of what the Western diet influence is through that cycle. By the way, every time they show a Western diet, they show a cheeseburger and fries. It's a lot more than that. It's actually, some of it looks really healthy on appearance, but it's not. So Western diet doesn't necessarily mean junk food like you get at a burger place. It could be a lot of things. So I wish we had more pictures to represent Western diet. A prudent diet in this paper might be what you see here, but all these factors are bringing in the fibers, the fermented foods, to sort of reverse, to add diversity and start reversing all the downstream effects that you saw earlier. So I'll break this down briefly for you and just say, let's dig into the food and see what is it in the food that is actually causing this influence or has this influence. All right, so I look at food kind of from a food science, nutrition perspective. Here are the big components. I'm not going to talk about fat, because fat is typically well absorbed. If it's not, you'll see it as fatty stool. It doesn't necessarily cause a major fluctuation in microbial sort of composition. But carbs do, protein does, and certainly fiber does. So I'll talk about that. Pretty quick survey here of really the spectrum of carbohydrates in the diet. They're classified using the degree of polymerization, so how many subunits there are in the carbohydrate, the bonding between those subunits, alpha bonds, beta bonds, that sort of describes what carbohydrate you have. And when you build the carbohydrate chains using these varieties of chains and alphas and betas, you get to alter the structure as well as the digestibility. So that's sort of the starting point. Nearly all short-chain carbohydrates are readily fermented in the by intestinal bacteria. So that's sort of, do we want that to happen or do we want to prevent that to happen? And there's, again, a balancing act between those two. And when they have a prebiotic effect, that's something we want to encourage, right? So this is just a quick drawing of how complicated this gets. There's all sorts of sugar, sort of carbohydrates that chain up in different conformations, different bond formations. Again, all this creates classes of carbohydrates. So monosaccharides, real quick, are really good for absorption, easy to absorb. Glucose and galactose are the most common ones. They have specific transporters. Fructose has its own way of getting into the body. These are monosaccharides again. One that depends on glucose and one that doesn't depend on glucose. At the end of the day, you want to absorb all your monosaccharides, and SCD is built around that. It sort of freely allows monosaccharides because they don't require digestion. There's no alpha or beta bond to break, so they're ready to absorb. And as long as you have an absorptive intestinal surface, you should be able to absorb them. Of course, you can overdo it and saturate it, but that's not part of SCD. Now, when you move beyond monosaccharides, you have polysaccharides. So there's a whole class, malto-oligosaccharides. These are the only polysaccharides that are actually digested in the small intestine. Humans can digest it. It's starch. Okay. Now, SCD excludes this, right? Rice, potatoes. But in the general healthy population, they should be able to digest these bonds and absorb them. But there are some that no human actually can digest, right? So galacto, fructo, you can see the list. These are undigested, remain intact, move down the GI tract, and serve as fermented food sources, energy sources for microbes. And so every time you eat a carbohydrate source, you're either feeding yourself or you're feeding your microbes or both. So it's a really interesting way to design diets to sort of supply the human with human food, and take it all in, leave no leftovers for the microbes, and then supply in your diet carbohydrates, fibers, that are not intended for the human, but are intended for the pets that we carry inside us. So this zoo that we have down there. And we're all zookeepers, right? We all have to manage that. We have to recognize it. There's things that we do as responsible zookeepers and the things that we're not responsible zookeepers. I know we've talked about sort of the garden analogy. That's a good analogy, sort of pruning and maintaining a nice garden. I think zoo might be a little bit more relevant because it's an animal sort of world out there, and plants can't really switch their energy source. Like you're either a photosynthesis light, and if you don't get the light, you die off in a garden, right? And a fungus might come and take over. But in us, the microbes can actually shift their energy sources. So if they're deprived of fiber, they'll go look for something else, right? It's the same organism, so it's a little bit less plant, maybe more animal sort of in nature. But you got to watch out. So what happens when there's a bit misbalance? So there's obviously soluble dietary fibers. I'll kind of go through this fairly quickly. And there's insoluble dietary fibers. And each of these are designed, and obviously Elaine in her book really dug into this and described which ones are legitimate, which ones are legal, which ones are going to be digested well in us and absorbed, leaving no food or energy for the microbes to shift them in the wrong direction or to deprive them. So now what is, kind of bringing it all together, it's becoming increasingly clear that a healthy microbiome, now we don't know what a healthy profile looks like this is the panel of a health, like blood glucose, here's the level you want to be at, right? We don't know what a healthy microbiome panel looks like. We just don't know. Like it's too early, maybe there'll never be one. But we do know that they all exhibit a wide diversity, right? Which I showed you earlier I didn't have. They exhibit stability, meaning they don't bounce around day to day, which I did. And they exhibit resistance, so you can sort of bounce back and not allow outside influences to modify you. And, you know, I'm not there. So diversity increases with a more complex diet. So increasing the range of dietary fibers is key, and we do that. So when I went off of a no-fiber diet to eventually SCD, to eventually late-stage SCD, I'm getting not just more fiber, I'm getting all tons of different chemical structures, alphas, betas, long string, short. So, and that's great because it's sort of that zoo analogy. I've got food for all sorts of animals in the zoo. Okay? I'm not, if I gave myself just a one single structure fiber, well, maybe that only supports one set of animals, right, in the zoo. I don't want that. I want a whole diverse zoo, right? Stability over a long period of time. This is an indicator of global microbial health. So you want it not to be bouncy so much, right? You can sort of go through days, weeks, seasons, and not see too much fluctuation. And resistance is a key characteristic, especially at time of significant life change. So for example, going through weaning, antibiotic treatment especially is a big thing, recovering from an illness. You want to resist all that outside influence. So if you eat out and there's, you know, a foodborne infection, somebody didn't cook the chicken all the way, right? Everybody else at the party is fine the next day, I'd be sick because I just didn't have that diverse resistant capacity. And that's what you want to avoid, is you want to have a vibrant, live, healthy population with a lot of energy to resist this insult, this outside sort of foodborne infection. All right, so all these fermented, these fibers that end up in the colon and the large intestine have beneficial byproducts, sort of end products of fermentation. And you've heard about this a little bit earlier. So short-chain fatty acids are a key down product or byproduct of fermentation. I'll go fairly quickly here. Proteins can also result in byproducts, which is negative mostly. But look at all the things that these short-chain fatty acids do for us. Okay, the acetic acid, propionic, butyric, they're source of energy for the gut itself, and they get absorbed into the circulation and do sort of service systemically. Now, protein fermentation has negative attributes. So a decreased fermentation of protein lowers the amount of ammonia and the amount of nitrogenous compounds in your system. In contrast, formation of protein fermented products have deleterious effects. So how does that translate to SCD? Well, protein should be digested and broken down in the stomach with the acid. The peptides should be broken down, the peptide bonds, and you should absorb all the amino acids, right? But proteins have a three-dimensional structure that's all onto their own. It's not chains of carbohydrates. Plant proteins typically have a very tightly wound amino acid three-dimensional structure. Really hard to sort of unwind and break down. And so plant proteins are harder to digest and may remain intact in the gut. So gluten is a plant protein. You got soy proteins, right? You go down the list. So I recommend, if you're having problems with protein digestion, to sort of shift and become a little bit more sort of carnivore based, so eggs and so on, until you're sort of able to process this amount of protein and then later shift to a more plant-based source of protein. I have people I support in the IBD community doing SCD as a nutrition clinic that I run in Seattle, and some people are doing great and they decide to become a vegan, and it sort of throws them into a spin because that large influx of plant protein is really hard to digest and break down, and it starts shifting what you see on the chart here. Okay? So something to think about. Now, this is kind of one of my favorite charts. This is how much fiber you have in your diet on the x-axis. So typically we don't eat enough fiber, right? I was hardly eating any. Average American, 15, I would guess. The recommended is about 20 to 25. And this, on this y-axis is the mass of the stool per day. All right, so this is just saying the more fiber you eat, the more you poop, right? But there's something else here. If you're taking in fermentable fibers, they don't produce as much stool. So where's it going? Here's an example. 30 grams of fiber produced 40 grams of stool. This one, more than twice. Where did it go? Well, it's fermentable. It got broken down, fermented, turned into short-chain fatty acids. Your body used it. It did all this good stuff for you. When you take non-fermentable fiber, it really doesn't have anywhere else to go other than add to your stool. So you want to balance the two of fermentable and non-fermentable, and that's sort of part of the design of how you want to mix your diet and foods and get a little bit of both. Now, before you do anything downstream on the diet, there was a little representation of this. This is sort of my visualization of an unhealthy gut that's just not able to hold on to anything, microbes, not absorb anything. It's just runoff, erosion, and we don't want that. We want to reverse that so we can actually plant and have a healthy mucus and get the right microbes to sort of engraft. This is a picture of, you might recognize this. This is microvilli on the epithelial surface, sort of the good version and not so good version. But this is different. This is not humans. This is actually squirrels who hibernate in the summer or not. So when they're actually hibernating, they're not eating, their epithelial sort of goes into silent mode, but it reverses. So this is kind of across all species, all mammals, you see this. And in this case, even in the hibernating state, the organism needed to retain its barrier function and its engagement with the microbiome. So it's a bit of a deeply rooted physiologic need. So knowing all this, how can we modulate the microbiome? What can we do to influence it to benefit us? We can obviously do negative things to it, but let's talk about some positive things. So here is a slide of fecal microbial transplant in IBD. The chart shows you where the donors were up here. This is the IBD patients before the transplant and after. So they actually pick up what was provided them, right? What was transplanted. But there's a little bit more to this story. There's a split. There's a subset of these post-FMT IBD patients that respond and some that don't respond. And it turns out the non-responders actually didn't really shift in their microbiome. They didn't really pick up what was offered, what was implanted. All right? And that represents the lower end of this curve. And the ones that did respond are the ones that were more closely matched with the donor. So it's a bit of a hit and miss. Am I going to take in this transplant? Am I going to show up as what the donor had, or am I not? We don't know yet. This is just another version of looking at it. So this is early stage in the FMT world. This is a small study out of Harvard. FMT may induce clinical remission in select populations through increased diversity. But they also caution this is early. We don't really know what the donor should look like, what the recipient should look like. And from my perspective, you may be able to inherit the donor's fecal microbial profile, but you can't inherit their immune system, because there's also the immune sort of sheriff in town watching over all this. And even more importantly, you don't inherit the donor's diet. You don't even know what their diet was, right? It's a kind of a sometimes mixed pool of fecal samples. So it's one thing to put in a population of microbes that were used to eating something in somebody else, and all of a sudden they have to adapt to your diet. And they may not last long. They may not benefit you. We don't know. So as this evolves, that aspect I think needs to be considered, is adjusting the recipient's diet to support the incoming microbes. And maybe SCD is a perfect remedy for that. Maybe you don't even need fecal microbial. So here's a couple more examples of what SCD looks like compared to a low residue diet, we see shifts in a positive direction with the microbial diversity. SCD had a bunch of species increase within 4 weeks. A low residual diet decrease because there were no prebiotic fibers. Now there's reviews on all this. There's several dietary interventions. Cochrane did a nice job reviewing it. I don't have time to go through this in detail, but the overall summary was they looked at 18 randomized controlled trials looking at diet intervention in IBD, 1,878 participants in active and inactive UC and CD, so pretty good, right? But they were all inadequately powered, so there wasn't enough statistics in the study to really make good interpretations out of it, conclusions. 14 out of the 18 studies had high risk of bias. So there's a real lack of knowledge, actually, in where we are with diet and IBD. All the studies were rated low or very low in terms of the certainty of evidence. We don't know. So these were all randomized control trials, which are the best we have in clinical research. But are they good enough? And I think they actually set the bar too high for, let's say, an SCD intervention study. All right? So you can't really be a placebo SCD person. You kind of know, right? And then the intervention has all these other variables along with it. So even if I introduce SCD to a cohort, I can't control exactly what they eat, even though I want to, I can put a dietitian, I can put a research team around it, but I can't control how they eat, right? How do they chew the food? Are they salivating or not? The enzymes are right there in your saliva. How fast do they eat? Do they get up and walk around right afterwards, or do they let the food digest, right? Those are the tons and tons of external variables that might influence the net benefit of any diet, and especially SCD, which is sensitive to digestion and absorption. So I actually don't think we'll ever have a real well-designed, sort of perfect gold standard study on SCD. I haven't done the calculation, but factoring in all these variables, you can overcome them by adding more people in the study. But if I do the math, I have a sense that the number needed will be more than everybody who's on SCD right now. It just doesn't add up. We can't do it. So the hurdle's too high. The clinical world, the GIs, the medical community, looks for RCTs of high quality with high power to sort of start convincing themselves. And I don't think it's going to happen. I think the bar's too high. I think the funding's not there. I think the interest isn't there really. So we're left with these sort of semi-good, semi-bad studies. You can sort of interpret them, and we have our community as evidence. So, I'm exploring how we can leverage that and build and consolidate that sort of crowdsourced evidence that's already in play in years. It probably won't satisfy the clinical community the way they want, but it's still evidence that I think is worth sharing and consolidating. All right, I'll skip some of these. These are the Cochrane studies. All right, so is SCD the best thing since sliced bread? All right, I'm just being funny here. But let's look at what the US government, the USDA, Health and Human Services, puts out as dietary guidelines for the general population for a healthy life. So these are what the recommended intakes of fruit are in the US, currently by the US government. The recommended range in blue for fruit for males and females on the right. The actual intake is in orange dots. Okay? So we're coming up short as a population in the US on fruit intake. Now, do you guys take about a cup equivalent of fruit a day? If so, you're at the average level. If you take 2 on SCD, you're right where you should be. Okay, so I'm postulating that SCD actually achieves the recommended intake requirements for a general healthy population. Here's fruit. Here's vegetables, right? Do you eat about, I don't know, I'm 52, so I should be about 3 cups of vegetables a day. Well, before I wasn't even eating any, right? If I'm at 3, I'm there, right where I should be. But on average, nobody in the US is. We're all missing the mark. What gives? Here's starchy vegetables. Even with all the potato consumption in the US, the average intake is below the recommended. I don't know why the recommended is so high, okay? Something like, I don't know, a large percent of vegetable intake is actually potatoes, potato chips, French fries, things like that. So we'll skip this one because it's really not part of SCD. Legumes, eventually this becomes part of SCD, right? Real big gap. It's actually a pretty high bar about what the recommended intake of legumes and beans are. 2 cups a day, including peas. So I bet you most of us, once we're later on in SCD, we're doing better than 1 cup a day. You know, add up the peas as well. Grains. All right, we obviously don't do grains here, but it looks like the US population is actually right where it needs to be. Right? Pretty good. But wait a minute. Grains have 2 flavors, processed and unprocessed, sort of whole and not whole. So once we split that, the average is good, but it's all because it's coming from sugar. Look at that. Refined grains, basically processed white flour, way over the requirement. Whole grains, way below. Of course, on average, it's pretty good. Okay, that's not what you want. Added sugars. Okay, we're way ahead of where the US population needs to be. Okay, we're, right? The US is way over the top. Okay, we're already ahead of the curve. Sodium. You know, the way we cut out processed foods, we also cut out processed added sodium. That's a big deal. There's no way I can figure out on SCD that you can get to the average US intake of sodium. Doesn't matter how much table salt you add. Okay? Because it's all coming from processed foods. So that's a big deal. Right? These are major health sort of policy factors on sugar, refined grains, sodium, vegetables, fruits. SCD's already there. So already SCD achieves the public health guidelines for a healthy diet. Why isn't everybody doing it? Okay, think about the implications. All right, let's look at quickly sort of the chart on sort of this complicated, well, which diet should I do, right? This is not even all of them. So there's Western diet, paleo, SCD. There's like, how can keto diet achieve its goal at the same time, low FODMAP? And there's evidence that it does. Like there's paleo actually, there's evidence that it achieves its goals. How can they both be right at the same time? Well, I kind of flip that argument and I say they can all be right because they have one thing in common, and they're all moving away from the Western diet. Okay? Simple. So I think roughly, if you move away from Western diet, everybody, no matter what health effects, obesity, diabetes, you gain 50% of the net benefit just by moving away from Western diet. The other 50% is like now tuned into what you need, what your flavor and palate and culinary needs and specific health needs are. And now you can sort of add the other 50% and you get to SCD, for example. And here's a chart comparing pretty much, you know, all the relevant diets. It's a little bit simplified, but it's got grains, fruits, vegetables, starches, and so on. Animals are pretty much accepted across all these diets except a plant-based diet, as you might imagine. SCDs over here. So, you know, red's not really allowed and green is. There's some squishiness there. But it's, in my mind, SCD is actually an expansive diet. Okay, this is one big misconception everybody has. Sure, early on, it's restrictive. And once it works, it becomes hugely expansive, especially for IBD patients who have already gone through a natural deselection of their foods. So here's a little mind map I use. I kind of reorganized all the food lists in Elaine's book and kind of categorized them according to logical groupings. Let me see if I can play this. Okay, here we go. So it's basically, you start with the baseline diet, broth, chicken, and as you grow, you just click on the categories and you get more vegetables, you get more classes, and they're all bundled according to sort of botanically relevant or agriculturally, chemistry sort of relevant categories. So, yeah, so all the legumes are together. So I want to somehow figure out a way to use this to help people not only visualize where they're at, so everything's shrunk down early on, and as they progress, you just click, and now all of a sudden legumes become available to you, and all of a sudden, this different class, mushrooms in different classes become available. And eventually, you get to a point where this map that started really small and shrunk down because it needed to, eventually becomes this massively expanded tree of ingredients that you can make almost an unlimited amount of recipes from, right? Seasonal adjustments, geographic adjustments, your own preference. All right, so it's almost an unlimited variety that you can add. So anyway, I just use this as a tool to kind of map and track who's where at any given time point. And of course, sometimes you may want to pare down, so you shrink it, hit the minus, and it goes away for a while, and then later on you catch up. All right, so real quick, oh, cool. Now, I want to talk about another personal experience I had on SCD. I showed you my data from last 3 years. This last April, my wife and daughters and we traveled. We went to Hong Kong, Shanghai, and Beijing for 3 weeks. And so I thought that would be an interesting analysis to do. So I ordered a bunch of fecal sample collection kits for myself, knowing everything we talked about earlier. And I collected samples twice in Seattle before we left, went on a 21-day trip and came back and collected 4 repeat samples afterwards. Okay, just wonder what would happen. I maintained SCD throughout, didn't do yogurt, that was just not part of the game. I did have 1 serving of rice at one point and 2 bread rolls. Other than that, I think I did a really good job. Took a lot of food with me, a lot of good restaurants, a lot of vegetables, seafood. No street food, a lot of good kind of restaurant, hotel food. Lost 3 pounds, but here we are. So let's see what happened. Now here are, these results just came 10 days ago, so I haven't fully analyzed them. But this is kingdom, phylum, all the way down to genus of this particular organism, Actinomyces. Right? I don't really know what it does yet, I haven't really dug into it, but it wasn't in me before. All right. Now it's still in small amounts. It might be good, it might be bad. I don't know where it came from. And it sort of persisted even at day 50. It sort of popped back in. This is Bifidobacterium, something we like, something we want. I'm low in it, right? Not good. But look, it came in, and it's sort of for the first time kind of enriched myself. How did that happen? What is this? Odorbia. I don't know what Odorbiacter. I have to look into this, but it was a big introduction that I didn't have before. And I have, here's Bacteroides, not necessarily a good one. On and on. So I'm diving into this, trying to explain it. Oh, here's a quiz. I took the name off of this one. So I had it, and then when I came back, it was gone. And then it slowly crept back in. What is it? Lactobacillus. It's my yogurt. Look at that. Yeah, I went off yogurt for 3 weeks. Okay, pretty cool. I'll skip a few of these. There's a whole range of these. And then I summarize them here. So this is the entire set. the 2 before and the 4 after, all stacked up. These are only ones that represent the total of 99%. So I'm showing you 99% of everything. And you can see up here, before Fusobacteria, it's not a really good organism. One strain of this has been associated with colon cancer. I gotta go look into that. I've been high before, I remain high. But look what happened during the trip. It got bumped down to the lowest level I've ever seen. And now these are all relative numbers, so the numbers may have been exactly the same, it may have even increased, but relative terms, it actually went down. And in came all those other species I showed you, all those phyla. So this is interesting. Now interesting also is after 50 days, it sort of started looking like what happened on day 0. So it's not persistent. Whatever happened didn't stick. I just went back to my home in Seattle, ate the same things, and kind of started looking like the before. Maybe a little bit more diverse. So now I'm still, interpreting all this. Travel's fun. I saw a bunch of fun things. I took this picture for Jeffrey. Okay, hotels had yogurt. It was actually quite sour and made on site, so I actually tried just a small bowl of it. And then you get signs like this. Okay. All right. So the lessons from this sort of journey adventure is SCD is compatible with travel if you plan ahead. Observed rapid shift in microbial community. You saw that. Even with a dietary adherence. So where did all the change come from? It's important to continue yogurt intake. All right. Stick with that. A few days maybe is okay, but regular. And then what other influences are there that might modulate or enrich the microbiome? And then something that opened my eyes, just geographic and people exposure. So in Seattle, I see, a couple dozen, 3 dozen people a day. It's maybe the same people. On this trip, I saw, 10,000 people by lunchtime. Right? Take the subway, go to the airport, take the train. So maybe that actually has a very positive influence, so you can actually think about building diversity through travel or unbuilding it by staying put. Up to you. I know it's hard, but think about it. How are we doing on time? Okay. I'm going to go through a quick side topic, which I want you guys to be aware of, because this is an up-and-coming field, expanding the influence of the gut microbiome beyond the gut itself, because there's many downstream influences beyond the gut. I'll just talk to you about a few of them just to sort of set the table for future discussions possibly. Now here are two mice, identical, implanted with a tumor, treated with a chemotherapy known to treat that tumor. All right? One mouse responds, tumor shrinks, one doesn't. But everything's identical, except one mouse had a fecal microbial transplant from a cancer patient who did respond, and one had a fecal microbial transplant from a cancer patient who did not respond to that same drug. Wow, right? So there's something going on, right? And we're looking at that, and the scientific field is looking into that. There's a whole angle around gut-brain axis. Your gut is connected to your brain, we know that. I won't go into too much detail, but the enteric nervous system, the brain, gastrointestinal tract, they're all connected. This is how we have bidirectional communication between, where immunity, digestion, metabolism, all these things are being back and forth communicated. Microbes actually produce signaling to the brain. This is how microbes communicate with the brain. None of this has been fully characterized. We've seen some animal studies showing some interesting interactions between gut and brain. There's interesting gut cells that we never knew existed. This is a recent finding called neuropod cells sitting in the gut, and they have a sensor for sugar. And once they pick up the sugar molecule, within milliseconds, your brain knows about it. There's actually a synapse formed with the vagal neurons that communicate the presence of sugar within milliseconds. So now the brain can make sense of what you're eating, at least what sugar you're eating. There's vagal signaling, so a wide-ranging role. This is the natural part of your physiology, to coordinate parasympathetic activity, regulate heart rate, gut motility. That's already in place. But microbes can influence that. So your gut microbes are actually producing neurotransmitters from the fibers they're eating. Right? This is another set of byproducts. So dopamine and noradipamine are produced by members of the bacillus family. They get absorbed and systemically distributed. GABA by bifidobacteria. Serotonin by Enterococcus. Right? They're producing it because they're symbiotic and evolution sort of selected for that, and they're having some positive influence. So these are the sort of the signaling molecules that we want to see. They also produce hormones and metabolites. So we talked about short-chain fatty acid metabolites, but there's also a whole slew of hormones. This is just a set of animal studies that are now looking at nervous system disorders. So we have Alzheimer's, ALS, and autism spectrum disorders, and responding to introduction of different prebiotics or microbes, which are now being called psychobiotics. So these are all animal studies, but this is a big burgeoning field. In this example, Alzheimer's disease was studied. Introduction of a specific strain of bifidobacterium reversed the impairment of mice going through a maze, indicating it prevented cognitive dysfunction. So there is a very interesting field, and maybe next year's conference we'll see more information maybe relevant to IBS perhaps, and maybe IBD. All right, so in summary, I talked about the environment influencing the microbiome. That's pretty much diet, but other factors like maybe travel influencing the microbiome, and the microbiome influencing your IBD state. And then your IBD state influencing your environment, like where you want to go, what you want to eat. So it's kind of a circular, but I could just as easily draw it this way, right? Environment influencing IBD, influencing right? Which one is it? These are all, none of these data I showed you are cause and effect, they're associations. And probably a better representation is this one. Okay? It's bidirectional, complicated. So if I had to summarize all the microbiome studies I've reviewed, everything affects everything. Just that's, if you want to conclude one thing, and if you don't believe me, look at this study. The effect of having Christmas dinner with the in-laws on gut microbiome composition. Actually, a study that was published recently. So they took people at Christmas, checked their microbiome before, and after they went to a family member's celebration, and then an in-laws member celebration. And guess what? There was an effect. When you go to an in-laws, you see a larger shift in your microbiome. When you go to your family, you don't see that much shift. Are we talking stress? Are we talking food? So the authors actually conclude, before we can recognize in-laws as a new environmental factor that can influence the gut microbiome composition and subsequently host health, a larger randomized control study is needed, and on and on and on. Wonderful, right? Who wants to sign up for that study? All right, I'm going to just sum up with a couple of sort of short, brief statements. It used to be in the nutrition field and the health field, it was always a good advice to just say moderation is key to health. You know, moderate what you eat, moderate exercise, all that. I think we need to change that a little bit. Modulation is key to health. Modulating the gut microbiome. Think about the things you're doing that are having an influence down there in your gut. The organisms, the profile, the diversity. Used to be that we say you are what you eat. Okay, in my case, I am what I ate yesterday. Okay, I'm bouncing around everywhere. And the goal ultimately for me is building resilience, not invincibility. So I'm not trying to do any of this to make myself Superman. I recognize that things will happen, but I'm building resilience. So I want to end with this quote. It's from one of my heroes. Actually, I went to boarding school in England when I was a child, and this is the most famous resident of that town, Charles Darwin. It's not the strongest of the species that survive, nor the most intelligent, but the one most responsive to change. So think about this, in terms of the microbiome, but think about it in terms of the SCD community. I'll read it again. It is not the strongest of the species that survive, not the most intelligent, but the one most responsive to change. Thank you.